Did you know that the average person has only 1 to 2 real friends they can confide in? Did you know that almost 43 million adults across the US age 45 or older are expected to be victims of chronic loneliness? And also, did you know that loneliness is alleged to be as dangerous as smoking 15 cigarettes a day? Loneliness is a global epidemic slowly making its way into the life of individuals. Let's get into the science behind this topic. As social mammals, we progress through life with the connections we make. Our brains push us to keep in company with others. Loneliness, another term for social isolation, is when one is deprived of this society. This can have profound effects on humans, such as depression, and in the most extreme cases, Alzheimer's. Scientists were uncertain about the precise location of loneliness until recently, when breakthrough findings have shown us the part of the brain. Loneliness actually stems from a part of our brain called the dorsal raphe nucleus. The DRN is located in the midbrain and pons, and the neurons in this area utilize many transmitters to control various physiological functions. Dorsal means the upper side of the human body. The raphe refers to the ridge that separates the two symmetrical parts of the body, and the nucleus refers to the cluster of nuclei in the brain. The raphe nuclei are the prime area in the brain for the production of the neurotransmitter serotonin, which is linked to helping regulate mood and social behavior. Lower levels of serotonin in humans have been associated with depression. MIT neuroscientists conducted a study on a group of mice to locate precisely which region of the brain represents feelings of loneliness. Each mouse was kept in isolation for 24 hours. This isolated group of mice displayed a strengthening of connections in the DRN. Another experiment placed a couple of mice in isolation and the other groups of mice together for 24 hours. The results showed that in the group of mice that were housed together, the DRN neurons were not heavily active. The mice that were in isolation for 24 hours displayed increased DRN activity and became much more sociable after the isolation period. These experiment results showed that the neurons in the DRN play an important role in the increased motivation to socialize with others. Studies have shown a correlation between smartphone usage and loneliness in teenagers. Teenagers tend to stay in their rooms and spend an average of 3 or more hours on their electronics daily. This group of kids have a 35% higher chance of a suicide risk factor and a 27% higher risk of depression. Loneliness is something that has serious effects, and it is not to be ignored. In today's world, people are attached to their smartphones, and this can cause major problems in the future. As humans, it is important that we collectively work together to combat loneliness. I'm Meghna Nair, and thank you for watching.